Ladies and gentlemen, thank you very much, and to Margaret for inviting me here today. It's not often that I have such an esteemed person to be a warm-up act for me. But since you mentioned Alder Hey, and in fact I've had it down here on my introductory slide, I should say that it's through Alder Hey that I have, and my interaction with people like Alan Milburn and Baroness Hayman and some of the others, that I've become even more motivated in favour of the use of human tissues. I'm not anti-vivisection, and I'm not against the use of animals in the appropriate circumstance in science. But what I am in favour of is when, as a clinician and as a scientist and as a translational pathologist, I am both of those, that we should provide the human species with the best evidence and develop medicines and approaches and therapies appropriate. Now, as a pathologist, most people think that we walk around in green aprons and Wellingtons and cut up the dead, and that can't be further from the truth. What we are, as pathologists, we are the advocates of the patients. And in fact, pathology having moved away into the so-called back rooms and the distant from the patient, pathologists are now coming very quickly to the fore in many countries. If I want to speak to my pathology colleagues in in Beijing, I come in the morning and I will call them at 7:30, 8 o'clock our time, 5 o'clock Beijing time. They are not sitting in their laboratories, they are with patients, discussing with patients the diagnosis and then what is going to happen to them. Because the pathologists in the Institute of Urology in Beijing and those looking after breast cancer patients, those are the ones who are doing the laboratory tests, which define how an individual patient ought to be most appropriately treated. In my laboratory, since 1988, down to now where we're producing, we have published, peer-reviewed, something in the region of 19 biomarkers, predominantly for use in human cancer, many in prostate, some in breast. As a histopathologist, we look down and we develop these markers, and I won't go through these in detail, except to say that these now tell us, and this was published only 2 months ago for the second time in the British Journal of Cancer now, we can tell when a man comes forward with prostate cancer which 1/3 of those men need urgent treatment and which 2/3 can be watched, so-called watchful waiting with impunity, because those 2/3 of men do not have aggressive disease that requires urgent treatment. So we can not only devote resources economically to the 1/3 that does, we can say with confidence to that patient who's come forward, yes, you have prostate cancer. No, it is not going to affect you badly. You do not need to take all your savings out of the bank, resign from your job, have chaos and upheaval in terms of your family. We will watch you with other biomarkers until such time as you need treating. Because however good treatment is, there are always side effects and risks. And it behoves us to manage the patients, not in terms of a prostate cancer, but in terms of, or something else, but as an entire human being. Why? If you take a man of 58, 55, let's say, with prostate cancer and you give him hormone treatment, he will develop a fracture of his femur within something like 10 years. If he has a slow-growing prostate cancer that does not need treating for over 15 years, you have shortened almost certainly that man's life by a considerable amount of time. It is much better to know the biology of the disease, to keep your hormone treatment until after a 10-year period and then treat him. Because otherwise, he fractures his leg, he's put into bed, he develops a pneumonia and he dies, whereas that was avoidable because one did not understand the disease. Here is another of the biomarkers in prostate. You can see the brown staining here, which is the invasive prostate cancer as opposed to the non-stained around. And we know by looking at the expression of markers such as this, and this is what was published recently, by looking at a large number of tissues through what we call tissue microarrays, where we take tiny samples of many of these things, we can put hundreds upon hundreds from individual patients on slides and look at them simultaneously. We have markers so that in the normal you get brown staining, the abnormal here is not stained, and these are the ones which are good. These are the good guys. That was the original finding for one of the biomarkers, that we could separate the 1/3 of men in red there who needed urgent treatment, 2/3 who did not express the particular protein. If you look at bladder cancers, these are different patients down here, the expression of different genes. The essence is that there are different patients who are going to respond differently. That type of information will not come from looking at animals. We understand if we give a particular drug, this is a network. This is part of the network of regulation inside a cancer cell. If we knock out a particular gene, we know from our studies looking at the human which genes are upregulated, which are downregulated, which of these now become the new targets. So that the essence is, by studying human diseases, we are now in a situation whereby we will treat a patient and we will be able to predict, if they respond, the next line of treatment that they're going to need and when it's going to be. And after that, the next that comes, simply by having studied the human cells and knowing those networks. So the essence is, if you really want to study human disease, you've got to study the human. Don't try studying something else as a surrogate. However tempting it might look because it's easier, you're going to get the wrong answer. Welcome to the future. The future is there. We know how we ought to take apart human diseases. And if you have been, thank you for listening.